This video will focus on free radicals, their generation, the damage they cause, and the body's defenses against them. Let's begin with a case report. A 2-month-old girl was admitted for persistent pneumonia and recurrent infections since her 6th day of life. Despite multiple courses of antibiotics, she had ongoing pneumonia. Examinations confirmed bilateral pulmonary consolidation and multifocal pneumonia. Laboratory tests showed raised liver enzymes, but normal immunoglobulin and complement levels. Critically, her neutrophil nitroblue tetrazolium, or NBT slide test, was negative, and she had abnormal neutrophil respiratory burst activity. What is the possible diagnosis and pathophysiology? The learning objectives of this video are to define free radicals and reactive oxygen species, understand their generation, the damage they cause, their clinical significance, and the scavenger systems that mitigate their effects. A free radical is an atom or molecule with an unpaired electron in its outermost orbital. This makes it highly reactive, with a very short half-life. It reacts indiscriminately to achieve stability by either accepting or donating an electron. Normally, oxygen is fully reduced to water. Incomplete reduction, however, produces reactive oxygen species, or ROS, such as superoxide and hydroxyl radicals. Hydrogen peroxide is a highly reactive ROS that produces other ROS, though it is not technically a radical. The tissue damage caused by these oxygen radicals is termed oxidative damage. This image illustrates some of the reactive oxygen species encountered in living cells. This slide outlines the various sources of reactive oxygen species within a cell. One major source is leakage from the electron transport chain in the mitochondria, where oxygen is sequentially reduced. Another source is the cytochrome P450 system located in the endoplasmic reticulum. ROS can also be generated during the degradation of purine nucleotides into uric acid. Interaction with transition metals can also lead to the generation of free radicals. Peroxisomes produce hydrogen peroxide during the Fad-dependent oxidation of long-chain fatty acids. This scene highlights the processes that lead to free radical generation. The respiratory burst is a critical process where neutrophils, eosinophils, monocytes, and macrophages produce superoxide anions via the NADPH oxidase enzyme during phagocytosis. The activation of macrophages drastically increases oxygen consumption, which is known as the respiratory burst, involving NADPH oxidase. This scene provides a visual representation of the NADPH oxidase complex involved in the respiratory burst. This slide shows a diagram of the components and steps involved in the production of superoxide by NADPH oxidase. Exogenous sources of free radicals include high doses of drugs like acetaminophen, which can cause liver damage, and peroxidation catalyzed by lipooxygenase. Radiation and light can also generate radicals, along with factors like cigarette smoking and environmental pollution. This slide provides a summary of the various mechanisms involved in the formation of free radicals. Free radicals can cause significant damage to proteins, induce lipid peroxidation, and harm nucleic acids. Cellular damage caused by ROS affects proteins, with amino acids like proline, histidine, and methionine being particularly susceptible. This can lead to protein fragmentation, aggregation, and cross-linking. In lipid peroxidation, the membrane lipids are susceptible to attack by free radicals, which compromises the integrity of the cell membrane. This diagram illustrates the process of lipid peroxidation, starting from a radical attack to the formation of lipid peroxyl radicals. Damage to DNA can cause heritable mutations in germline cells or contribute to cancers in somatic cells. Products of lipid peroxidation, such as dialdehydes, can also modify DNA bases and cause chain breaks. This visual summarizes the various ways radicals cause tissue damage in the body. Free radicals can play a role in autoimmune diseases by modifying proteins. When these modified proteins are recognized as non-self, antibodies can be produced that cross-react with normal tissue proteins, initiating autoimmune responses. The total body radical burden can be estimated by measuring products of lipid peroxidation using assays like the ferrous oxidation in xylenol or FOX assay, estimation of dialdehydes, or measurement of pentane and methane in exhaled air. The FOX assay measures lipid peroxides. These peroxides oxidize ferrous iron, which then forms a chromophore with xylenol orange under acidic conditions. The TBARS assay measures dialdehydes, like malondialdehyde, formed from lipid peroxides. These react with thiobarbituric acid to create a red fluorescent product. In the pentane and ethane assay, the peroxidation of N6 polyunsaturated fatty acids forms pentane, while N3 polyunsaturated fatty acid peroxidation forms ethane. Both can be measured in exhaled air. The clinical significance of free radicals includes their role in chronic inflammation, such as rheumatoid arthritis, and acute inflammation, where macrophages release them. ROROS also induce tissue damage in conditions like ulcerative colitis and chronic glomerulonephritis. In chronic granulomatous disease, or CGD, the NADPH oxidase enzyme is absent. This means bacteria can be ingested normally, but cannot be effectively destroyed, leading to persistent infections. In respiratory diseases, breathing 100% oxygen for over 24 hours can cause lung edema and endothelial destruction due to free radicals. In premature newborns, prolonged high oxygen exposure can lead to bronchopulmonary dysplasia, or ARDS, in adults. In the eyes, free radicals are implicated in retrolental fibroplasia, or ROP, in premature infants treated with pure oxygen, causing vascular injury. Cataracts are also related to aging and photochemical generation of free radicals. In atherosclerosis and myocardial infarction, LDL deposited in endothelial cells is oxidized by free radicals. This attracts macrophages, forming foam cells and leading to the development of atherosclerotic plaques. For skin diseases, plant products like psoralens, combined with UV light irradiation, produce singlet oxygen to treat conditions such as psoriasis and leukoderma, providing a clinical benefit. In carcinogenesis and its treatment, cancer therapy uses radiotherapy, which produces reactive oxygen species in cells to trigger cell death. This image highlights the critical role of antioxidants in combating free radical damage. Antioxidants protect against cellular injury by scavenging ROS and other oxidants. In a healthy state, there's a balance between free radicals and antioxidants. We can also supplement these antioxidants externally. Antioxidants can be broadly classified into two categories: enzymes like SOD, catalase, and glutathione peroxidase, and non-enzymes, which include various proteins, vitamins, and other compounds like transferrin, Vit-E, and Vit-C. Superoxide dismutase, or SOD, is present in all oxygen-metabolizing cells. It converts superoxide radical and hydrogen to hydrogen peroxide and oxygen. SOD contains different metal cofactors like manganese, copper, and zinc. Mitochondrial SOD is manganese-dependent, and a defect in this is associated with idiopathic cardiomyopathy and premature aging. Cytoplasmic SOD is copper and zinc-dependent, and a defect here is linked to amyotrophic lateral sclerosis. SOD concentration increases during exercise because aerobic metabolism increases the generation of ROS. This, in turn, stimulates the cell to synthesize more enzymes, leading to increased protection during post-exercise or rest periods.